All right, so good morning. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much. My name is Dr. Ariel Pradipta. Thank you very much, Dr. Mutia, for the very warm welcome. I've been told by my students that I talk a lot, so I'm going to set a timer for myself. Because the 30 minutes is actually need to be split between myself and the distinguished Mr. Himawan over there, because we need to deliver a very interesting journey. So today, I will try to display the potentials and the journey, the difficulties, and the heartfelt moments of being able to utilize long-read sequencing for investigations for disease mechanisms. Now, the challenge here is for us to define disease mechanisms. So I'm going to tell a story that came from my own PhD journey when I was studying in Osaka University. So I was told by my sensei, Yamamoto sensei, his name was, a very unique gentleman and a very smart scientist, that there's two ways to think about immunopathology, because that was my PhD on. And the immunopathology can be thought from a thinking process that starts from structure, and then we go into the different function and the dynamics of each of those functions. Now, the other way is that each of those structures in immunology especially, and later on we will see in genomics, rarely is involved in only one dynamic. They are involved in multiple dynamics, and each of those dynamics lead to several plot twists. Now, this occurs in both host immunology and pathogen immune response. This also happens if you are, I'm sure you are all very adept at reading all the different genomics and whole genome sequencing studies, genome-wide array studies, and all those different things. We will see that different dynamics can relate to several functions, especially diseases. Now, in a simplified way that totally undermined my 5 years of blood, sweat, and tears in PhD, we begin by looking at two very important gene sequences, which is the IRGs and the GBPs. Now, both of them are important in recognizing intracellular pathogens. And then through the magic of CRISPR and the magic of genome editing, we created, I was assigned to create a knockout mice. So I knocked out that specific IRGM2, and I found out that the mice that doesn't have that gene structure, basically led to their death after they're infected by a certain pathogen compared to wild type. So obviously, that certain gene is very important, and whatever protein that comes with it. So we had to find out what was the function. At this point, we know that the function is to keep the mice alive after an infection. But what's the dynamics behind that? To put it simply, this particular gene produces a protein that attaches to a vacuole that comes inside your cells after they're infected by a certain parasite or bacteria. And then they allow the body to decide, the innate immune system, to decide whether we should kill the parasite or that this is a host cell, that it does not need to be killed. So the gene itself, the protein that is produced by the gene itself, does not lead to the killing. It only leads to recognition on whether this is friends or foe. Now, when we think about it that way, there's a very simplified way of doing it. So after I try to mutate, I try to make different kinds of that protein with changing different sections and name them all these wonderful numbers, M77A. That's actually the name of the amino acid position and the change that I did. And I found out that by modifying those specific parts of that structure leads to different activities. One mutation doesn't allow parasite killing. The other mutation still allows killing, but it doesn't allow antigen presentation. The most severe one doesn't allow everything. It doesn't allow antigen presentation, and doesn't allow parasite killing. And all of these kinds of explanation is now trendified, being made very knowledge by this one term that everybody keeps saying, omics. It's like everything now ends with an omic. There's a transcriptomic, there's a metabolomics, and there's genomics. Now, all of this is just part of a figure, part of the journey that we want to figure out disease mechanisms. And those disease mechanisms, again, trying to remind us, comes from both analysis of upstream and downstream. Now, how we view that tends to be very different when you're a clinician or when you're a molecular biologist, when you're a microbiologist or when you're a clinical pathologist, when you're a pathology anatomist, or whether you're involved in oncology. The upstream and the downstream are very, very different. But we are lucky because this is part of a big journey to realize dream to reality that started way back in 2002 and accelerated very quickly from 2004. In 2002 was the first announcement of the genome-wide array study, the GWAS. And then later on, in 2005, is the emergence of next-generation sequencing. Now, next-generation sequencing allowed us to high-throughputly to analyze the sequence of the DNA and the downstream activities in a more rapid way. Then, we need to discuss about what's the analysis and how we connect genotype to phenotype and all that. And I will tell us all about that challenge on another lecture, because I only have 7 minutes left. Right. So in this case, all of this is part of the dream to reality that is, it's kind of realized, and it kind of, I wouldn't say realized, but precipitated in 2017 when it was the first case report of gene therapy for sickle cell, anemia, hemophilia, and the first human testing of immunotherapy against sequencing-defined patient-specific neoantigens, which is Bu Ines have pleasantly introduced to us using the terms precision medicine. Now, these are the kind of things that are accelerated by next-generation sequencing and the ability to do long-read sequencing for the completeness of the reads. Now, this is something that relates to me because I use glasses and I have myopia. And the kinds of analysis that comes from there allows us to find that there are genes, for example, sorry, this is too small, you're welcome to find the paper later, but there are genes that relates to how myopia progresses. And all of this came from next-generation sequencing. Now, to discover disease mechanisms, they actually had to sequence in animals too and find homologs of those genes in animals and in humans and try to discover, this is in animals, they make animals have glasses and then they try to cure their myopia by taking off the glasses. And they found that there are different genes that are overactivated or underactivated from a transcriptomics point of view. And from an ophthalmological insight, we saw that this is, again, part of a very long journey of detection. For example, from 1869, the first description of congenital amaurosis, which is, unfortunately, blindness. After 131 years, through the development of all these things, there's a successful gene therapy for Leber's congenital amaurosis in dogs. All right? So, but now the clinical trial is open, and I think you can check it out in clinicaltrials.gov. Now, all of that is part of the journey of how we had to basically find out about these disease mechanisms. And turning science fiction into science is made possible because there is groundbreaking treatments that are allowed right now, that are enabled to the many different analyses that we can do. Now, delivering these discoveries, a whole different story. And to describe this without taking the thunder out of our next wonderful speakers, I will talk about one that relates close to immunology, which is methylation and epigenetics. Now, a lot of methods have been done to analyze epigenetics and methylation, but one of the most versatile ones is using long read because it allows for non-modified analysis. Now, there are two ways, there's two very important journeys that I would like to introduce today. The first one is from the structural molecular biology, in which they found, through methylation analysis, a section, the green one over there, the unique section, that is very important in nucleosome remodeling. That is a very structural approach. It is not about phenotype that comes later, but it is a structural genetic analysis. Now, it actually relates to aging, because one of the most important mechanisms of aging is genomic instability that is aggravated or alleviated based on their epigenetic alterations. So these are the kinds of disease mechanisms, and I guess in terms of aging, it's not much of a disease. But as again, apparently we found a 104 years old just now that was mentioned by Bu Ines. I swear I didn't plan this, Bu Ines, but apparently it becomes very relevant because these are the kinds of things that we can discover right now. On the other side of it, from a diagnostic point of view, one of the most important findings in terms of using long read is the diagnostics for meningioma. Because WHO grading now have adopted the use of methylation status for them to grade the meningiomas. And these are the different kinds of analysis that are needed to complete this grading based on the new WHO staging. I think there's some attendance from PON, and I think I cooperate mostly with Dr. Yuli and Dr. Cynthia. I hope they're here and very excited to see this. And with a lot of this development leads to a very conducive environment to talk about the use of epigenetics analysis for discovering disease mechanisms. Now, the downstream of that, or maybe upstream of it, again, depends on how you look at it, is the different ways that we can analyze transcripts. Because we want to know not just from the gene itself, but also the expression and the protein existence later on. There are several steps in the middle that I simplify, but I simply do not have the credits to talk about it today. And so in terms of transcriptomics, this is an interesting one because NGS transcriptomics, next generation sequencing for transcriptomics, allows for high throughput analysis on the many different mRNAs and RNAs that needs to be analyzed through a certain sample, for example. Now, of course, there are strengths and weaknesses, and all of this require discussion. And a lot of this can be seen on the versatility of it from a publication in Nature, I think around 2 years ago, 2022, that showed the robustness of analysis for transcriptomics using next-generation long-read sequencing. Now, this dynamics of progress can only be enabled if we are able to look at it from I use a very basic concept that I think we're all very aware of, host, environment, and pathogen. So I want to see that the host is the lab solutions that we all have right now, that we're all excited to develop. And then the pathogen is the vision, while the environment is the ecosystem that we're building right now in this room. So the lab solutions include so many different things to be able to use it, to understand it, and to analyze and to synthesize all those different explanations. And then the ecosystem is literally the discussions that we have right now, the different collaborative efforts that hopefully can create something that is greater than the sum of its parts. And the target is to understand the different diseases that I'm sure later Dr. Tip will talk about applications in the medical school, and then Prof. Gunadi will talk about applications in rare disease, and I think Dr. Ira also will talk about that. And hopefully we will be able to lead to collaborative genomics, where we publish all these different journals and being able to collaborate with the different people around. And just on time. Because to reach that stage of collaborative genomics, as you can see, we have that lab environment, the ecosystem itself, and the ways that we have to build this ecosystem. And because of the many different diseases that we're involved right now, ranging from cancer, rare diseases, ophthalmological diseases, even infectious disease and immunology, it requires an end-to-end discussion. A place to be able to discuss about various solutions that we need to ask and answer and discuss. And with that, I would like to invite Himawan to be able to lead us in this discussion, because he will talk about how Genomica will be able to provide these solutions for you.